This is Group 9 presenting the findings of a 2018 study by Burns et al. Variants in XOSC9 disrupt the RNA exosome and result in cerebellar atrophy with spinal motor neuronopathy.
Before diving into the findings of this study, we must first explore some background. The exosome is a 9-subunit complex involved in mRNA processing and degradation. Recessive variants of the exosome XOSC3, XOSC8, and RBM7 have been associated with conditions affecting the central nervous system, particularly pontocerebellar hypoplasia type 1, which is investigated in detail in this study. An examination of morpholino knockdown of exosome components in zebrafish presented similar disease phenotypes as seen in affected humans.
The researchers hypothesized that mutations in RRP45 cause deficiencies in the exosome that result in defects in neurological development.
In this study, fibroblast cell cultures of homozygous variants XOSC9, 3, 8, and RBM7 were grown in high glucose medium with fetal bovine serum and antibiotics. As shown to the right, affected individuals presented symptoms of cavulvarous feet, joint contractures, and fisted hands with adducted thumbs. The common notable signs of disease for all 4 individuals were prominent developmental delays, muscle weakness, motor neuronopathy, and respiratory impairment paired with progressive cerebellar atrophy seen in brain MRIs of the individuals.
The following techniques were used to analyze the genes of each of the 4 individuals in the study. Chromosomal microarrays used both copy number and single nucleotide polymorphism probes to identify mutations in the genome. Exome sequencing helped reveal the specific variants in each of the individuals through sequencing of coding regions in the XOSC9 gene. Sanger sequencing determined the exact DNA code in these regions of mutation, and haplotype analysis uncovered a common 80 kilobase haplotype among all 4 individuals. The figure on the right shows electropherogram results for the 2 identified XOSC9 variants at amino acid positions 14 and 161. Genetic analyses identified 2 types of variations in XOSC9 among the 4 individuals studied. Individuals 1, 3, and 4 were homozygous for the C41TC p leucine 14 proline variant, whereas individual 2 uniquely had compound heterozygosity of the C41TC p leucine 14 proline variant along with the C481CT p arginine 161 nonsense mutant. 4 pedigrees are depicted representing each of the individual cases. Individual 2 with the compound heterozygosity experienced more severe defects resulting in death.
Researchers immunoblotted the muscle extracts from the second affected individual to visualize the effects that the compound heterozygosity of the 2 variants in XOSC9 had on protein expression. In this gel, we can see the almost complete absence of the XOSC9 band compared to the presence of the band in the 4 controls from unaffected individuals. Alpha-tubulin was used as a loading control in this experiment, and subsequent gels shown in this presentation will use actin or GAPDH as loading controls. These indicate that the samples were properly run on the gel, and the absence of the bands for the exosome component of interest corresponds to severely decreased expression of that protein in the samples from affected individuals. Fibroblasts from individuals with a mutation in either RBM7 or XOSC3, 8, or 9 were then immunoblotted for all 4 of those proteins. This had the more predictable result of showing that the mutations corresponded to decreased expression of the affected genes. However, researchers also found that they corresponded to decreased expression of other exosome components. They noted that these effects were particularly significant for XOSC9 expression in all variants, XOSC8 expression in the XOSC3 variant, and XOSC3 expression in the XOSC8 variant. They then used blue native polyacrylamide gel electrophoresis, or BN-PAGE, of protein lysates from the fibroblast samples to isolate and visualize expression of the exosome complex. This showed that variants in one component of the exosome corresponded to decreased assembly of the entire exosome. Then they performed RNA sequencing of the total RNA in the fibroblast cell lines of control and affected individuals. This allowed them to identify differentially expressed genes and the resulting up or down regulation of associated function correlated to the XOSC9 variants. Up until this point, the exosome had been linked mainly to the degradation of AU-rich transcripts, but researchers suggest that the widespread effects of XOSC9 variants on the overall transcriptome show that the exosome is likely involved in processes beyond this sole function. This may include splicing or other gene expression regulation.
In order to demonstrate that the loss of function of XOSC9 caused the common phenotypes in the 4 patients, the authors generated 2 zebrafish models. The first one used a morpholino oligonucleotide to block the 5' splice site for a 146 base pair intron in XOSC9. This mutant is known as the morphin. This caused the retention of the intron, resulting in a frame shift and the mistranslation of the protein sequence. This is visible as 2 different bands in figure 5b. Increasing amounts of morpholino resulted in increasing amounts of the larger transcript with the retained introns. On the other hand, the second model used a guide RNA to cause Cas9 to cut within the targeted region in exon 3. This resulted in frameshift mutations being introduced. This mutant is referred to as the crispin. In Figure 5c, the authors show one frameshift mutation involving a deletion of 5 nucleotides within the targeted region. In 5d, the authors demonstrate a correlation between the proportion of mutagenized cells in the zebrafish and the degree of the phenotype mimicking the common phenotype in the patients.
This variety in how severely the embryo were affected by SOSC9 knockdown were classified into categories of normal, mild, moderate, and severe as shown in Figure 6a. The morphin were zebrafish that have a morpholino introduced, and crispin were zebrafish that were subject to CRISP or Cas9-mediated knockdown. Researchers now classify mildly affected embryo as one with smaller head and eyes. Severely affected embryo have smaller than normal eye or no eye at all, very small head and truncated body. As you guys can see at the Figure 6b, so that compare relative distribution of morphological phenotype in SOSC9 morphin and crispin.
Whole mount immunofluorescence of zebrafish embryos was used to investigate whether XOSC9 was required for brain and neuromuscular development. Whole mount refers to a staining technique where small pieces of tissue, usually embryos, are stained without sectioning, which preserves the three-dimensional spatial information, allowing for a comprehensive interpretation. Immunofluorescence involves using a primary and sometimes secondary antibody, which are chemically labeled with fluorescent dyes to visualize the distribution of a target molecule throughout a sample. These images show whole mount immunofluorescence on XOSC9 morphins and crispins with an antibody against the neuronal marker HUC. As you can see in the morphin and crispin, the midbrain is misshapen and the cerebellum and hindbrain are completely absent, compared with the uninjected control, indicating that the brain fails to properly develop without XOSC9.
In Figure B, whole mount immunofluorescence was done on islet 1 GFP transgenic line of zebrafish, which produces green motor neurons in the hindbrain. Cranial nerve V is split into two distinct parts, anterior VA and posterior VP. In the XOSC9 porphins and crispins, it was common for VP to be absent. This again suggests that functional XOSC9 is required for the posterior part of the brain to develop in zebrafish. Figure C shows whole mount immunofluorescence of synaptic vesicle 2 shown in green and alpha bungarotoxin shown in red. SV2 is with motor neurons and alpha bungarotoxin is with neuromuscular junctions. The whole mount immunofluorescence showed that motor neurons and neuromuscular junctions failed to properly develop in XOSC9 morphins and crispins in comparison to the uninjected controls. In Figure D, Phelodian staining was done. This is where the actin filament is stained, showing muscles. Phelodian staining showed that muscles failed to develop properly as fibers were sparser, more spread out, and irregular in the XOSC9 morphins and crispins. Together, these results show that XOSC9 is important in neuromuscular development.
In conclusion, researchers found that autosomal recessive variants in XOSC9 are associated with early onset progressive axonal motor neuronopathy, resulting in severe weakness and respiratory impairment in combination with cerebellar atrophy. Exosomopathies, a collection of rare diseases caused by mutations in XOSC genes, share the common features of spinal motor neuronopathy and cerebellar atrophy. Results suggested that the exosome complex cannot be assembled or might be unstable if one unit is primarily reduced, and that disease manifestations and severities are a result of degree of overall exosome dysfunction rather than individual exosome component dysfunction. Additionally, altered gene expression in XOSC9 variants suggests that exosomes may have a more widespread function, perhaps in splicing or regulation of gene expression. Lastly, immunofluorescence in zebrafish models demonstrated how exosomopathies affect neuromuscular development and showed how zebrafish can be a useful tool for in vivo studies of RNA processing disorders. Some outstanding questions we had were, what are some other examples of exosomopathies, and how do mutations in other exosome components cause similarities and differences in pathologies? How might the exosome be involved more broadly in gene expression? Does it play a role in splicing? How can the zebrafish in vivo model be used to study other rare genetic diseases of the brain and spinal cord? And lastly, how can we further study neuronal cells of affected individuals or zebrafish neurons to reveal neuron-specific alterations?